up to this point, we've looked at different kinds of models, additive models, interaction models, some basics of multi-level models, and we've fit some different models to the same set of data. But what we really haven't done yet is try to figure out which of those models is the best model, right? By some definition of best, and we'll see there's lots of possible definitions there. And that's what we're going to do in this unit, is to talk about model comparison. We're going to, in this lecture, we're just doing an introduction to that, kind of why this matters, and sort of what we're trying to balance here, and then we're going to go into some specific techniques for model comparison. And so the challenge that we're facing is one of underfitting versus overfitting. And what this means is, do we have kind of too many predictors or not enough predictors? And ultimately, we're going to sort of take the stance that if we have fewer assumptions, meaning fewer predictors are preferred, so we want to have a model that's good enough but not too good. And then we have to kind of, with definitions for preferred or best model, we have to figure out how to compare them. We have to sort of figure out how good is good enough, meaning, how good a predictor, needs to be. All of these things are going to be sort of wrapped up together in this whole process that we're going to go through. And so really there's this trade-off, right? So underfitting, like I mentioned, you'll frequently read this as bias, especially if you're reading the machine learning literature. I find that term terribly confusing in this context. Underfitting, I think, is kind of self-evident, right? The model doesn't describe the observed data well enough, right? And that leads to poor prediction. Overfitting is the opposite of that. This is variance. Again, we use variance in lots of other different contexts, and so I find that, again, in this context, it's not helpful. But again, if you read the bias-variance trade-off, that's really what people are talking about. Overfitting is a model that has learned too much. It describes the observed sample too well, and that leads to poor prediction. And you see both of these actually lead to poor prediction. And so what we want to do is find the model that leads to the best prediction. And so we really need to balance overfitting and underfitting, or underfitting and overfitting. And so one way to sort of think about this in your mind, if it helps, is when you fit a model to data, right? That model is effectively learning something about the observed data, right? So it's learning how to relate the observed data to the outcome variable. We can put more stuff into the predictors. That lets it learn more. Right? But we can put a lot more in there, and maybe it doesn't do as well in the end, right? We could put not enough in, and that leads to underfitting. And so what we want to do is find this balance of what we call regular features, so things that we are, information we're interested in, and then the noise, the irregular features. So we want to sort of ignore the noise and learn from the parts that we want to learn from. And this is very abstract at first, but if you can kind of sort of think about all of the things that we'll be doing from here on as trying to sort of meet this balance, then that will sort of help you understand them conceptually. Maybe less so in the kind of mechanics of how to do them, but that should be more straightforward, the mechanics.
Yeah, so, any modeling that you do, there's often this, you can think of models as not just statistical models, but theoretical models too, as falling on kind of a continuum to, more general versus more specific, and often that goes along with, being less realistic and more realistic as well. The more realistic your model gets, the more, features you include in it, the more specific it often gets to a smaller and smaller, set of problems that you're trying to understand. And so that can be at play here too when you're trying to balance these two things, is that as you add predictors into a model, you might be getting better and better at explaining your very particular collected set of data, but that's not going to really let you learn about the more general biological problem that you're probably trying to answer. And so, that's just one of the things that goes into this balance. Okay, so why are we making models in the first place? We've already talked about this a little bit in terms of, needing a way to make sense of our data sets and what's going on and make conclusions about the biology involved. And we've talked a bit already about, the goals that we have in doing that in terms of estimating parameters and things like that, and also making decisions, making conclusions, hypothesis testing. And it's good to remember that no model that we fit, even the most specific and realistic model, is going to encompass everything. And so none of the models are going to really represent the true biological process. That is a good thing, because that is something that is so complex that we can't really totally understand it.
And if we did actually know the true biological process, we wouldn't need a model for it.
Yes, that's right. We wouldn't need to
we would
understand
just use the process, right?
Yeah, that's right. Yeah, sometimes models are. Hana Koko has a book about modeling, and she makes this analogy that making a model is kind of like making a map to a location, and that a map that is totally realistic is really just a representation of reality, right? You're standing on the street. That's a totally realistic map. You can look around, and it's not informative in terms of telling you where you need to go. And so we have to have these simplifications that we call models that sort of pull out the most important features for our goal, for our question that we're focused on.
All right, so we're going to work through a relatively simplistic example in a what is probably a strange way, actually, but it'll hopefully the point we're getting to will make sense here. And so what we're going to use is a very small data set with just 7 data points, where we have the estimated, measured brain volume from the cranial endocast, and the estimated body mass from 7 hominins. These are genus Homo. You see there number 7 there is Homo sapiens, that's humans. And obviously, again, like we always say, looking at lists of numbers is not informative. So here's what the data look like.
Here's Homo sapiens.
There's Homo sapiens up there, and so we related brain volume to body mass. And it looks like there's some sort of positive relationship here, which is not surprising, right? You could think of just like physics.
Yeah.
Bigger body should have a bigger head, and a bigger head should have a bigger brain. And so it's nice to work with a small data set like this, because what we're going to do is fit sequentially more complicated polynomials to these data.
Yes, we're going to try to describe the relationship between body mass and brain volume in more and more complex ways.
Yeah, basically allowing more flexibility in the relationship here. And so these are, we've basically got all 6 of them here. This first one, you see this can essentially is, m to the first, so just m. So this is just a straight line linear model. And then m squared, and m cubed, et cetera, et cetera, et cetera, and down to here where we have all 6 of them in a 6th order polynomial. And we're going to use R squared, which we learned about to compare these models. And this is a thing you could do.
Metric.
Yeah, it's metric, and this is a thing we could do, right? It's not, we're using it for teaching purposes, but people have actually done similar things to data, not unlike this.
Yeah, and you should sort of apply this example as you're thinking about adding what would be different predictors. Here, right. All of these are dependent on mass, but you can imagine, an experiment where you're adding more and more factors, maybe environmental factors, for example, to explain what you're seeing.
Yeah. All right, so the simplest one, we'll just sort of run through these relatively quickly. This sort of straight line linear model has an R squared of about 0.48. So that means we can explain about 48% of the variation in brain volume by this intercept plus straight line single slope model of body mass.
Yeah, and so that's what you see here, right? We're just explaining this positive relationship. The line's constrained to be straight here because of our model.
Yeah, and the bands here represent the confidence interval on this line, and you can see that this is that Homo sapiens out there. That by this model we have much bigger brains than we should for our body mass. All right, so let's keep going. Here's the squared term. R squared is now 0.53. That's what this looks like. You might wonder about this model because now it's actually predicting that as we go further and further out, bigger hominins, we're going to actually get smaller brains.
Eventually.
And that the largest brain should actually be associated with a body mass of maybe about 47 kilograms. Does that make sense? It's a cubic power. Now we're at 0.67. The other thing that you'll notice as we sort of keep adding predictors is that this line is going to get closer and closer to all of our observed values. We're up to 0.82, 83 now. The fourth power starting to take on some really strange shapes. We have a maximum now. Actually, as we get bigger, brain volume is gonna move off to infinity, and as we get smaller, it's also going to move to infinity.
That's starting to track the points pretty well.
Yeah, we get to fifth order now. We're about 99% of the variation is explained. You can see we actually fall really close to the line. There's a few little dips and bobs here.
Yeah, so remember with R squared, if all the points fall in the line, right, then that means the model can explain all of the variation. There's no unexplained variation is when you have, residual variation, when the points are distant from your model fit in some way.
All right, and we do the sixth order, and now essentially we've explained all of the variation. Right, R squared is 1. There is no black shaded line now because our points are perfectly encoded by the sixth order polynomial. And you can see we had, as you remember, we started with 7 data points. We have an intercept plus 6 more. That means we have essentially 7 estimated parameters, and thus we have all of our points perfectly predicted by our model.
No degrees of freedom.
No degrees of freedom are left.
You can see this very strange relationship where we're predicting that first brain volume is going to increase, and then as you get bigger, it's going to go way down and then come back up again, right? So obviously not very biologically realistic.
Right. But again, this is not something you would do, but you might keep adding more and more and more predictors. And as you do that, you will see that your R squared goes up. And that's what we can see here. It goes up from 0.48, about half the variation, to 100% of the variation. All right, so then we might sort of think about, just without kind of putting any analysis on it, which of these models is the kind of best balance for under versus overfitting. Right? We could have a model we didn't even fit with a mean only, right, where we predict that every species
the best estimate is the mean for everybody.
Mean. Everyone has the same mean, right? That's probably really poor in sample prediction, meaning in sample meaning the observed data that we have, and probably really bad for data that we don't have, what we call out of sample prediction. Model 1 might be good. It looks like, it kind of fits with what we know about, the basics of geometry. So that might be good. Model 6, clearly there's too much information there. But maybe Model 2, we don't know, right? This is what we need. We need explicit ways to actually kind of answer these questions.
Yep. Yeah, so this is a quote from McElrath. We won't read it to you. You can pause and read. But essentially the idea here is that, fitting a model to your data is easy, but predicting out of sample is difficult, right? So, you might imagine in the last example, you might collect a new hominin and add it to your data set, and if you have overfit your model, you won't predict that new observation very well. And typically the biological questions that you're asking, you might not necessarily be explicitly thinking about prediction, but usually we're trying to make a general conclusion, meaning that we, want our model to inform data that's not in hand. Right?
Yeah, so if we were to collect new data,
yeah,
does it also fit with the model?
Right. Does this apply more broadly in biology?
And you might be aware of this kind of existential crisis in science of not being repeatable, and that's probably partly related to this, is if models are fit and basically learn the data too well, then they don't generalize to other new data.
Mm-hmm. So we have different methods for model comparison that we're going to go through. This isn't an exhaustive list, but these are the ones that we're going to cover. So the first one is likelihood ratio tests, and we'll talk a bit about information criteria, and then we'll talk about cross-validation.
These are the next 3 lectures.
Yep. So that's what you have to look forward to, and enjoy.